Can you hear me? It's all absolutely mutual. It's been a blast. Best move I've made. Very welcoming. Some of my students are here. I love my students. My evaluations, I never had a class say, he loves this class. It's like, but it was totally true. And I really have. Can we get front lights down so the screen is easier to see? That's the one request. Get that.
And I'll push the wrong button, so
No,
just
it's all right.
bear with me.
Yeah, it's right. Right. Perfect. Excellent.
Is that okay?
You got it. Okay, so we'll go with this, and I'm going to talk about mathematics and molecular medicine today. And I'm really grateful to all my colleagues and everyone at the College of Science and Health in the math department and the University of Utah Valley University community, students especially, for their interest in these things. It's been really fun. And I'm going to start out with a little math problem that will come in later. It's sort of one of these classic problems that you may have played with. I'm thinking of a positive number. Half of my number times one more than half of my number is equal to twice my number. What's the number I'm thinking of? And later on in the talk, the answer will actually have some medical, clinical significance. But I'll come back to it, so don't miss the talk for that. So what is molecular medicine? There are various terms for this, or personalized medicine sometimes. Diagnostic methods and therapies. A lot of people talk about molecular diagnostics, but I find just as interesting molecular therapeutics. So what if they can tell you, you've got this, can they do anything about it because of that? And it's methods and therapies, diagnostics and therapies that target specific molecules, DNA specifically, one of the specific molecules that are crucial to healthy cellular function. When it's your DNA, you want healthy cellular function. So I'll say, whose DNA? It's not just always your DNA. It might be the pathogen's DNA. Then you want unhealthy cellular function for the thing that you've got. And so what is DNA? I said targeting specific molecules. One of the crazy things about DNA, deoxyribonucleic acid, sounds like a chemical formula, sounds like some chemical, right, with some fixed chemical formula. But it's actually not a chemical like H2O or C2H5OH or NH3 or today's talk I highly recommend by Bill McKibben on CO2. All of those things have a specific molecular formula. But the chemical structure of DNA changes. What the actual number of carbon atoms and things will change because the number of certain bases in it that make it up will change from DNA molecule to molecule. So DNA isn't just DNA, is kind of my point. But I'm going to give you a way to think about DNA that actually captures a lot of the useful things, because mathematicians like to have a simplified model of things in their head. And so just sort of come up with this and checked it with my med school folks, and they say, okay, it's kind of okay. DNA is the medium on which the programs that comprise the common operating system of every one of your cells is stored. Everybody has the same DNA in every cell in their body. And it's the operating system. There are different things that make it run differently in different cells, but it's a medium on which programs are written. And so it's a little bit like a CD-ROM or a DVD-R. But a couple of things where the analogy goes a little further. It's a backed up set of double-sided CD-ROMs. And one side is printed in one direction, one is in the other. That's not like CD-ROMs. But DNA, one of the first things I learned about DNA that I wasn't familiar with was it has a direction. You don't go down the two strands in the same direction. One goes this way, the other one goes that way, okay? So think of it as a backed up set of double-sided CD-ROMs. You get 2 copies of each disk. Those are usually called chromosomes, and those contain parts of the DNA. So the whole operating system is put into about 23 disks, and you get 2 copies. And usually when you get your operating system, the 2 copies are the same, but your 2 copies of DNA are mostly the same, but it gives nature a chance to experiment with different codes in different copies. So sometimes everyone usually has 2 of the same program for most of cellular function. But every once in a while, someone gets a different attempt, a different experimental variant in one of their programs. And sometimes someone gets 2 variants, and that can either be an interesting thing or it could be a health problem, okay? So the main programs in this operating system are called genes, and they are the programs that give the recipe to make proteins that are used in cellular function, okay? But that's not a bad analogy, I don't think. I've gotten reasonable feedback, and it's a double-sided. Each CD-ROM is double-sided. I say CD-ROM specifically because you want your DNA to be read-only. You don't want to change the operating system too much in your cells. But just like in a CD-ROM, you take it too close to a big electromagnetic field or some radiation, you might have a problem with one of your programs getting mutated. Okay, so, and the opposite side is a mirror image that is printed in the other direction. One direction, opposite side, and the mirror image thing. So basically, if you know one side, you should be able to replicate the other side. Okay, that's something. And in cells, what they used to do to diagnose if you had some infection, they would put some of your blood in a dish with some nutrients and replicate, let cell division go and reproduce the DNA. And it would take days, and it might be contaminated, all kinds of things. So the old days, you took DNA and you basically replicated the whole cell, including the DNA, all the DNA, even though you really only cared about a particular part. And so in the 1980s, Kary Mullis got the Nobel Prize for finding a way to basically do replication of just specific parts of your DNA. Basically copying the cellular mechanism of the DNA splitting into 2 complementary pieces and letting the other side of the copies be made in the test tube so that you would get, if you had 1 copy to begin with, you would get 2 copies after 1 cycle. You do it again, you get 4 copies. You get 8 copies. There's exponential growth. So here's what's called PCR, is replication in a test tube. And it's an interesting thing. You need one of the proteins the cell makes, called polymerase, to catalyze the replication of the opposite strand of DNA, the opposite side of the CD. But to separate the DNA, to split apart the sides of the CD-ROM, you have to heat it up. Imagine your CD-ROM, you heat it up, the 2 back-to-back sides peel apart. That's what happens to DNA, and we'll go into a lot more detail about that. But what happens when you heat it up to peel it apart, the thing that rebuilds it would get degraded, no longer work. So to do artificial cell replication at the beginning, when Kary Mullis wrote the paper, you had to do 1 cycle, you had to add this catalyst again. Do another cycle, add this catalyst again. And someone very clever said, gee, wouldn't it be nice if we didn't have to replace the catalyst and open up the thing and get contamination every time? It would be really nice, but you'd need a polymerase, a catalyst, that could withstand very high temperatures. Where might we find that? Well, we might find that in living things that live at very high temperatures. Where'd they go? They went up to Yellowstone. They went up to Yellowstone and they found a polymerase in a algae kind of thing that lived up in the Yellowstone hot pools. And they found polymerase that was thermostable at high temperatures. And ever since, that's what has been used to make this work. Okay. So PCR, polymerase, is the catalyst that allows replication. PCR, polymerase chain reaction. When you say chain reaction, it just means exponential growth. 1 leads to 2, leads to 4, leads to 8, just like a chain reaction in fission. Neutron splits an atom, you get 2 neutrons, split 2 more atoms, split 4, etc. Okay, So, and now here we're going to get to a little math. We're about to the point where we'll get to some math. You can get exponential growth, but at some point in your test tube, you're going to run out of the raw material to replicate your DNA. It's going to be harder for the stuff that's trying to find new things not to just come back and find its old other strand. So instead of exponential growth going on forever, at some point it levels off. And we call that logistic growth. And we study that in Math 1100 and Math 1220. So these are topics modeling exponentials. We even model exponentials in 1050, which I'm teaching now and taught in the fall. The number of cycles, the number of copies of your DNA. So what they wanted to do was see, is their DNA being replicated? That's detection. Is this specific piece of DNA in there? Is hepatitis C in there? Is mycobacteria in there? Do you have this infection? Is there exponential growth going on that's targeted to the specific molecule? How would you know whether you've got a lot of DNA in there? You add a dye that finds double-stranded DNA, finds the CDs that are put back together, and for each amount of CD that's put back together, it will glow when you shine some light on it. These dyes are green. And so this is my colleague, by the way, Carl Whitwer, Randy Rasmussen, who co-founded a company that built some of the first thermocyclers called Idaho Technology. And those things, the light cyclers that do this, that watch the DNA replicate, appeared in the pathology TV show House, except it was in a place where you'd never be using it. So we have the picture of it in the lab there. Anyway, so the idea is, if you have dye in your reaction, the brightness of the fluorescence can be used to tell how much DNA has been copied. If nothing comes up, it says, ooh, that thing wasn't there. If it comes up bright the same brightness 2 cycles earlier, you probably had about 4 times as much stuff in there to begin with. That's the intuition. And so here's the quantification, actually, and a dilution series where you take less and less and less of the target, and it takes longer and longer for the fluorescence to grow. So fluorescence should be approximately proportional to the amount of DNA and the amount of the initial target. So this is how you quantify DNA targets that you're looking for. Okay, but it's going to get a bit more sophisticated. So what happens? You raise and lower the temperature. What this is indicating, you're raising and lowering the temperature. What's happening? That's called thermocycling. You raise the temperature, the CD peels apart. The DNA unwinds. You lower the temperature, you put in parts of DNA that you know the sequence of, and it will find its brother and sister on the other side, either side, and start rebuilding. Just like real DNA would in a cell, but just the part that you've targeted out specifically. And you do this over and over again, and I might even try and go back and say, that's what's happening. All that's happening is you're raising the temperature, lowering the temperature, raising the temperature, lowering the temperature, raising the temperature, lowering the temperature. You have all the stuff in there to begin with, and what you start seeing is fluorescence coming up. And if you had twice as much stuff, it comes up 1 cycle sooner, and then follows the same pattern. That's quantitative PCR, called qPCR. How much detection is there any of that specific DNA sequence in your test tube? qPCR is how much was in there, actually. Okay? And it's based on the fact that the dye gives fluorescence proportional to the amount of double-stranded DNA. So there's your amplification, and there's detection and quantification. If your curve had been like this, what does that mean? Nothing was there. There was none of the specific thing you were looking for, which you identify by putting the specific pieces of DNA that you know are around the region you're looking for to start rebuilding it. Those are called primers. So you have to pick out the particular part of DNA by knowing exactly what you're getting. And you can actually call up the oligo house, Bob Shackman, oligo house, and you say, I want A, C, A, G, G, T, T's, whatever the bases of DNA are. You call this sequence, about 20 of them, and they'll give you enough milligrams or micrograms of tons of just that little DNA, and they will start the replication just on the right part of the DNA for just the right gene or organism. Okay. And so, detection and quantification. I've told you a little about what's molecular medicine. What's mathematics? I kind of think math is the study of relationships, because physics is the study of physical relationships, energy, motion, mass. Chemistry is chemical relationships. I think these economics are financial relationships, biology are relationships involving life. Math is the study of relationships in themselves. Inferential relationships are logic. Geometric relationships are shape relationships. Symbolic relationships, algebra. Rate of change relationships, calculus. So I think what makes math useful in so many things is it's the essential study of the relationships that you want to study in particular cases, and why the same math might work in physics and in chemistry and in biology and in economics and in social sciences and in the development of arts. And the other thing that's universal, what makes science science, is the reproducible experiment. Isn't our college about health and science is about the scientific method, which is the reproducible experiment? What in mathematics encapsulates the whole idea of the reproducible experiment? A function, one input, same output. That's what a reproducible experiment is. So I think that's part of why math is useful. And how is it used in this particular case? Well, I want to model that exponential growth of bacteria or that bit of DNA in PCR. So say this is something my 1050 students should be able to do, if some of them from my semester are here, and we actually just did this. I want to fit my growth curve by an exponential because it's doubling, 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 but it might not be exactly doubling. So I want to fit it by the population is the initial population e to the RT. There's a sneaky way I teach that says, what if you had 2 fluorescence units after 1 cycle and 14 fluorescence units after 4 cycles? In other words, you've grown by a factor of 7 in 3 cycles. Well, I want to fit the points 1, 2, and 4, 14. And here's some, bear with me for a slide or two of math. There's the answer. I can just write it down in terms of my data. 1, 2, 4, 14. I say, I start with my 2, I put that there, I put the ratio, 14 over 2, which is my 7, and I put t minus 1 there, and I put 4 minus 1 there. So you can see where the numbers came from. Okay? Is that right? Is that right? Can I do it off the top of my head? That's something, don't you have to write down 2 equations and 2 unknowns and solve them? No. Why does that work? What happens when I put in t equals 1? What happens to this fraction in the exponent? 1 minus 1. 0. What's anything to the 0? 1. 2 times 1, 2. If I put in 4 instead, I get 4 minus 1 over 4 minus 1. What's anything over itself? 1. What's 2 times 14 over 2? 14. There you go. Okay? And you can always change it to base e if you want by saying 7 is e to the log 7. There you go. So I don't have to solve it. I can do that off the top of my head. What that shows is I can fit any 2 points by an exponential. Remember that. I can fit any 2 values by an exponential, and I can actually do it as a party trick if I want. I bet you I can fit your exponential in my head. Okay? And so the kind of problems we teach, I don't want that. The kind of problems we teach in 1050, I say there's really not too much you can do once you have an exponential model. You can say, well, what will the population be after time t? Just plug in. What was the population, or what should it have been to reach a certain thing? Well, just solve for p0. Okay. What else? I have 4 variables: the future value, the current value, the rate, and the time. The other things I can do are how long till it reaches p of t, or what was the growth rate? I just divide and I get e to the RT is p of t over p0, and I take a log. I can solve either way. I think it helps because there's so many kinds of problems that you get in these things. You say, wow, how many kinds of problems do I have to solve? Well, there's just so much you can do with this formula. You can solve for this, you can solve for this, you can solve for this, and you can solve for this. And all the problems fit into that. And it turns out to be very useful. Okay, but now I want to be more specific. After I amplify, we do something called melting analysis. We do a slow temperature rise. And we watch the fluorescence go down very carefully as the fluorescence as the temperature increases slowly, the DNA unwinds. The CD-ROMs, double-sided, peel apart. And the exact sequence on it, you can read the sequence by how slowly it peels, because it's a zipper with triple tooth zippers and double tooth zippers. The CG bonds are triple bonds, and the AT bonds are double bonds. And if you did it really slowly, you could actually see AT is just broke apart, and CG, not quite like that, but pretty much like that. Okay, so what you're seeing is the fluorescence decrease as the temperature rises, and what I'm going to show you is the changes in that thing called a melting curve can be used to detect minute changes in the DNA sequence that have biological relevance. Okay, but it's not as, you need math to make it work. You need some math and you need some technology. So now we have a new reason to amplify, not to just see if hepatitis C is there, ARUP's biggest diagnostic, but which particular species of hepatitis C, because the different types respond differently to different treatments. Okay, so you can analyze specific mutations, a C to A mutation, or a C to T mutation, or a C to C mutation. Or a C to G mutation, what? When I got into this, I was so dumb. I thought a CA mutation meant on the two sides of the double helix, you had a mismatch. You had a C here, and instead of your CG, you had a new base in there, like a CA. But that's not what this means. Remember, you have two copies of each CD? It means on one you have a CG, and the other you have an AT here. So that's why you can have a C to G, because your CG became a GC. Okay? When you say a mutation, DNA is always complementary in the cells. DNA is always complementary in the cells, but not in the test tube, and I'll show you why now. But when you say a mutation, it just means it's not that you're making it not complementary. It means that you got a different pair on one as on the other. In other words, the program, they experimented with the program in your other backup copy of your operating system. Kind of a working thing. Let me try and show you how in the test tube, hopefully. Yes, I want to VGA input. VGA input. Yeah, that should do it. Okay, here's a little animation of how I do get mismatches in the test tube. Because say I had now here's a little terminology, is wild type means the most common normal DNA. And here I'll even focus on the mutation site. My animator friend Dale Meyer helped me develop this. So you have CG on both copies at this point in the program, on both of your copies. And then let's have another pair of copies of the DNA. And I'll just go back. I'm going to heat it up. They separate. Did you see the melting curve when it's separated? That's a melting curve for CG. Okay. Now I'm going to mix it up. I'm going to cool it down. There's nowhere else for it to go. They always end up complementary. So everything's the same. I heat it up again. I get the exact same curve. Now what it means to have a mutation, I'm going to show you where it's mutated in both. So notice you had CG. I'm going to have it mutation in both. That means, both CGs changes to ATs. And what happens if I focus on the mutation? See, that's now AT. Now if I heat it up, I get a melting curve. And I cool it down. They all mix up. And I heat again. No change, right? And here was my original melting curve. was over there. Okay? And it's a little bit apart, isn't it? Can you see the difference? Hopefully. They're slightly apart. Very close, but slightly apart. And that's the hard part to detect. Now, when it's called heterozygous, that means, oh, when I have a C to A mutation, this C became an A on the other pair, on the other CD. Okay, now what's going to happen? When I heat it up, not when it's being rebuilt, but when I just heat it up and let it cool back down together, oops, there's where you got trouble. The A on one is now mismatched with a G. They got together, but only one base was different. But that causes a lot more instability. So when I heat it up, this is the sort of crazy thing that one mutation on one double strand of your DNA is easier to detect than the other. Because what do I have? I have now my same two. I have the AT, okay. I have my CG, okay. But I have these two, and they melt at much lower temperatures. That's this one. There's a much lower duplex. There's a much lower. And what you see is the weighted average, something I talked about in the math classes, of all four. And that's very easy to distinguish from the two normal, wild type, and heterozygous mutants. So what you measure in practice, you can see these two, this is much easier to distinguish. And I'm going to come back and show you a way to try and separate these out artificially. So what you want to detect is the DNA normal? Is it heterozygous? Is there a difference between the two copies of the CDs? Or is it actually different on both copies? That's basically the big question of diagnostics. Is the program normal on both copies? Is it normal in one but not the other? Or is it abnormal in both? That's the biggest question in DNA diagnostics. So now I'll go back to the computer. No, these. There. Okay. So now, here's how you can't do this with the old dyes that detected whether DNA was there. It was a dye called cyber green, and these were their melting curves. And look how noisy they are. The dye doesn't stick to the DNA proportionally. So if I had any different species of hepatitis B, which is the blue one, I guess, they'd all look about the same. They tried to find other dyes that saturated the DNA better and gave a higher resolution curve, but it inhibited the whole chemical reaction. You couldn't replicate. So the big discovery in the last 10 or so years, it's about 10 years ago, it was when I got into the field, they found a dye that was actually much higher density that didn't inhibit the reaction. That's when high resolution melting became possible. It was the discovery of a dye called LC green, that allowed you to get much more smooth, high precision curves than cyber, that didn't inhibit the reaction. I think that was the beginning of high resolution melting. And that's the dream of high resolution melting. Could I do replicates of every kind of DNA with the heterozygous mutations, very easy to distinguish from normal, because they all have that inflection point, but actually each different type of mutation. I can tell, and even the homozygotes. So this is the idea of, these are actual melting curves of every possible mutation at one site. And it's two replicates, 2 or 3 replicates of each. And can you tell the difference between each of these groups of curves? I think you can, and a computer can. So can you tell the difference between the TT, the blue ones, and the green ones, and the red ones, and the black ones, and all the different colors there? Yes. That's ideal high-resolution melting. Okay. But here's the reality. That's what the melting curves come at you looking like. You say, can I tell the difference between all of those? I can't. And here's where math came in again. So what they tried to do was remove background, and they put a straight line here and a straight line here, and they looked at the difference between those lines. That's called the baseline method. Unfortunately, it just didn't work. So here's where it talks, see, fit to two two-state model with sloping baselines, and you take the subtraction. Well, that was what everybody did, and it didn't work. You didn't even get baselines that didn't cross before. That's a picture of the method that was used to separate out the background from the thing. And so I was told by my colleague that the dye itself decreases fluorescence with temperature exponentially. And so I said, wait a minute, why are you fitting lines? Why not fit an exponential? Okay. And so what do we teach in Math 1100? That the sum of the slopes is the slope of the sum is the sum of the slopes. Okay. I know your melting curve is flat here and here, so the entire slope is attributable to the exponential background. Okay? All slope is dye background, and it's exponential. We also learn that the derivative of an exponential is an exponential. So the background is an exponential, and all the slope is coming from that background exponential slope. So there's the picture. There's the raw fluorescence that you see messes everything up. I know the signal I'm looking for is this. The background has the same slope. This slope is this slope, and this slope is this slope. Because the slope of the exponential is an exponential, I can fit it to two slopes. And that's a unique thing about exponentials that you can't do with polynomials. You always need at least one amplitude. My mathematicians here know, fitting by a polynomial, you can't fit a polynomial uniquely to just slopes. But exponentials, you can. And that's what happens. Now you get, using the exponential background removal, with math that we teach here in our classes, I can separate out the background from the signal, and there's my signal. There's the normal, there's the heterozygous, there's the homozygous, and I can tell, okay? And where is that used? All over the place in medicine. So there's the exponential background method, and it was so useful that it became patented and licensed, okay? There's melting curve analysis with exponential background subtraction, and there's the entire formulas right in the actual patent publication. And there's nothing other than Math 1050 and Math 1100 stuff in there. So pay attention to your classes, and you can do some stuff with this. Now, patenting math, well, Google patented PageRank, and Stanford got 336 million, so math is very involved, but the patent office, someone in the patent office really hates math, had a really bad experience. And he says, we won't count, to practice before the patent office, we won't count courses in the history of science, engineering, field identification, independent study courses, high school courses, mathematics courses, one-day conferences, what? Wait, high school courses, mathematics courses, they really didn't like math, so I wrote a letter in protest of that, and they may be changing it. Anyway, so back to getting the colors. How did we get the colors? We used clustering methods, okay? And clustering is an algorithm for deciding similarity, which things are more similar than others. And here are multiply aligning sequences of hepatitis C to see which species, which type of hepatitis C are in. These are actual sequences from ARUP, and we did multiple alignment and phylogenetic trees. And actually here, Keith Ogden's lab is kind of world leaders in developing better methods for doing multiple alignments and visualizing them with phylogenetic trees. Seems to be out in the nice parts of the world with his students doing biology and bioinformatics. And you can see here's just a little piece of the hepatitis C 5' untranslated region. See all those ACGTs? That takes the algorithms that I got to talk to Keith Ogden about to get better things, because the way we're doing it's pretty hard. Okay. What else could we do? I said in the abstract, transplant compatibility. You can either try and test your siblings to see if they're transplant compatible, and it takes $1,200 per sibling, and it takes a couple of days, and you may need that transplant a lot faster. You may be lucky to live in Utah where you have 11 siblings, but you still got to test them, and that's going to cost a lot. So this is actually a Utah genetically heavily studied group, but it's a much more economical thing to do is look at everybody's melting curves. It only takes about $5 and 2 hours to amplify and melt, and to look at the siblings, that's not 4 curves, that's all the curves. Using exponential background subtraction, I can look at a particular part that's the lymphocyte antigen, the immune system, things that determines your transplant compatibility. And I see, oh, which siblings are compatible? 9, 10, and 16, 3, 5, 6, 11, and 17. They can all transplant among each other. And you can back it up, and they all come out the same. So this is actually probably more accurate than sequencing. And it's cheaper, and it's faster. Okay? And what you're not finding what type it is, you're just finding are these things the same type. And again, here at UVU, Daniel Fairbanks here, real genetics expert, Glendon Parker, student, are using proteins to decide whether 2 sets are the same and to do identity testing. Not identify particularly, but are they the same? So lots of great work in these fields going on here. At Idaho Technology, they have an FDA-approved diagnostic that uses high-resolution DNA melting to decide whether you've got any of these respiratory pathogens very fast. And coronavirus, influenza, enterovirus, parainfluenza, coronavirus, it will diagnose these using exactly this melting technique. And this method was actually developed by Liz Doman-Copine, who's now at Idaho Technology, project manager there on high-resolution melting. When she was in my calculus class about 10 or so years ago, she got 100 on the final. I said, "What's your major?" She said, "I don't know." I said, "Well, it better be math." And she went on in math from 100 in calculus in her final, got her PhD, and now is working in high-resolution melting and developing really great diagnostic tests. Okay, this can be used for early cancer detection. Here's melting curves and selective amplification to find, it's hard to see up there, 0.01% mutant allele fraction. You can see the cancer cell at 1 in 10,000 using high-resolution melting analysis and see that it's there early. So early cancer detection is another thing, and that's got a patent application also. How do we design these assays? We do theoretical thermodynamic prediction depending on the sequence using nearest neighbor Ising models, okay? And we have web applications to predict, given the sequence, what does the melting curve look like and where does it separate earliest, okay? And that's the UMelt application that's talked about in this paper. There are additional things called snapback primers that can get around the heteroduplex issues, and those are called snapback primers. Adam Millington from Orem, who's here today, has developed a better way of predicting a very small parameter model that took the melting temperature errors down from 10 degrees C average, maximum 30 degrees C by other models. He got the errors down by a factor of 10 to 1 average and 3 max. Okay, and what does that look like? That looks like that. And Adam came to my calculus class last year as an architecture major with a background in musical theater, asked me if I could help him get started programming, and the rest is history. He's now a member of our lab doing great work in primer design. Okay, and here are the models, a little bit more about the thermodynamic models. I'm going to kind of go through this quickly, but certain mutations, you can predict it. I always loved this term, in silico. You learned about in vitro and in vivo. It was in the dish or in the body. In the computer. Let's just do life in the computer, and that's in silico. And there's our prediction, and there's the actual. They're pretty good. And what we use, it's not just CG, GC content, AT content. It's whether a CG is next to an AT. So you break it down to your CG next to your GC. This is actually a specific, let's see, where is it? This actually is related to hypercoagulability. More clotting than you want or less clotting than you want. There are a lot of studies for those mutations that cause clotting disorders. And what you find out, you add up entropy and enthalpy for each of the nearest neighbors, and you plug it into what's called the Van't Hoff dissociation equation. That's over there. Van 't Hoff got the first Nobel Prize in chemistry, by the way, and developed this model that we still use for melting curve analysis at the coarser level, okay? And the homozygous mutant is indistinguishable because of a symmetry in the sequence. A C changes to a G to a C, and it's surrounded by a TA symmetry also. In that case, the nearest neighbors, when you add them up, are the same whether you look at it this way without the mutation or this way with the mutation. So it doesn't change. And there's about 4% of 5 million, I analyzed 5 million SNPs, single nucleotide, single base changes that cause disease in the human genome. 4% have that symmetry, and you need a better way to distinguish those. And the temperature change is 0, theoretically and practically. The single base, the single pair change is much easier to distinguish. Okay, and so what can you do? Can heteroduplex formation be our friend in resolving? And the method is I'm going to add extra wild type, extra normal to every sample. When I add extra normal to normal, nothing changes. If I do 50-50 normal and doubly abnormal, suddenly I've got an artificial het. The mutant and the wild type are the same. So my mute curve has all of a sudden gone all the way over. Really easy to distinguish. But unfortunately, my het curve has become almost more like a wild type curve, and I can't distinguish it. And that's what everybody was doing. They were adding a 50-50. The mute all of a sudden is over here, but now it's hard to distinguish from the het. So you replaced one indistinguishability for another. And so what do? You look at the number of those mismatched duplexes, and you say, aha, wild type, add stuff, nothing. Start with mutant, there's nothing, but then if you get to 50-50, you got all this. But I've only brought this down to here, and it's the separation of these curves becomes the thing that tells me how distinguishable are all three curves. So it's a weird calculus problem. It's a maximum difference between three curves, or a maximum minimum difference between three curves, okay? And you find the answer, and it's at x equals 1/7. Okay? 1/7? Is there a better reason? And this works, and they use that. They add 1/7. They have 1/7 mixture extra wild type. The reason it's 1/7 is 6 to 1. 1/7 is 6 to 1. If I split my 6 into 2, wild type and mute, and add 1 to one of them, I get 4 times 3 is 12, is twice my original 6. That was the problem that I asked at the beginning. What number, 6, if I multiply half of it, 3, by half of it plus 1, do I get twice exactly what I got? That's the optimal separation. That goes kind of fast, but I can give you the reference to the paper. What happens if you have more than 2 copies of your DNA? 2 copies of your CDs. So rather than 1 backup copy, bananas have 3 copies. Peanuts have 4 copies. Wheat has 6 copies. It's called the ploidy, so you call wheat hexaploid. Strawberries are octoploid. Octopuses are not octoploid, though. Okay? And how many genotypes do you get? It's a kind of a nice exercise. How many different types do you have if you have different number of changes of base? Well, it turns out to be Pascal's triangle on its side. You turn your head, 1, 3, 3, missing the 1, 1, 4, 6, 4, 1, 5, 10, 10, there's Pascal's triangle. Okay. And so you get lots of pictures and you do it numerically. It's a lot easier to figure out what the best mixing fraction is in all these different cases. Make pretty curves. Okay, the last 3 slides are about cancer diagnostics. You can look at how many copies of the gene you're called for. In other words, how many times do you run a certain program on the CD? And if that's broken, the number of times you call the program, if it's too much or too little, that causes problems in the cells, including cancer. And what we found were some mutations using some mathematics, again, that found 3 genes that were connected to a transition, just like HIV, between a dormant disease and an aggressive cancer, a dormant cancer and aggressive cancer. And we looked at how many copies of each gene, of 6,000 genes, were changed between the dormant people, the same person, normal and aggressive. And when we interrupted those 3 genes that we found mathematically, lymphoma growth was inhibited in mice. So it actually not only gave a diagnostic, but it gave a way to actually treat the tumors and stop the tumor progression. And these are microarrays. They're really just matrices of numbers. Which gene and which patient. And you can do some melting curves to quantify. And so what we found is if I made a graph in 3 dimensions of all the sets of 3 genes out of 6,000, that was a lot of numbers. How many choices of 3 are there in 6,000? There's an exercise for you. And analyzed what in any 1 or any 2 they overlapped, how many copies were being called for. But I found 1 set of 3 in the whole possibilities of 3 out of 6,000 where there were 3 genes where these were the dormant expression levels, how many copies of the gene you called for, and here were the aggressive. And look, wow, you can see, wow, that's what's changing. And I showed this to Kojo Olenitoba-Johnson, a hemopathologist, and I said, this is what the math says. These are the only 3 genes that do something. And he said, that's interesting, Bob. This gene, the x-axis, says the accelerator pedal is stuck. Proliferation is overexpressed. He said, the y-axis going down, he says, that's the brakes are broken. Apoptosis, cell death, is underexpressed. Your accelerator pedal is broken. Your accelerator pedal is stuck. Your brakes are broken. What's the third? I said, what's the third gene? He says, you can't take it out of neutral. The translation is overexpressed. It's going haywire. And he says, and those 3 are all on this same pathway, and we can interrupt it. And that's how this turned into a tumor suppressive therapy that can be used for B-cell lymphoma. So again, math in that. We're using similar methods for breast cancer in a new diagnostic for whether breast cancer is going to recur. And that's also going up against the patent office. And also here, I hear all the time down my hallway, Tim Doyle has also been working on breast cancer and actually using physics, ultrasound to detect the difference between different kinds of tumor tissue and do ultrasound for intraoperative margin assessments in breast cancer, breast conservation surgery. So, and he's always involving his students on this. So that's why I'm really glad to be here, because my colleagues are all as eager to involve students, and my students are great and want to get involved and doing great things, interdisciplinary things, joining things like our College of Science and Health, which are together to put together math and physics and biology and health sciences. And I think there's a lot of things to do on the horizon in new advances in this, so it's not all over. And I want to thank all my colleagues that I've been working on this with, Whitwer Lab and ARUP, Canon Life Sciences, Idaho Technology, and a lot of these people in Whitwer Lab at UU Pathology. Thanks.